Here we have our neuron, our sodium, our synaptic cleft, our sarcolemma, and our muscle cell. The action potential, which is the stimulus, releases an electrical current which travels down the neuron. The calcium goes into the neuron. Inside the neuron is the acetylcholine, and when the calcium goes into the neuron, it pushes the acetylcholine down through the neuron into the synaptic cleft. Acetylcholine leaves the neuron into the synaptic cleft. The acetylcholine travels across the synaptic cleft and the sarcolemma and opens the gates to the muscle cell. Once the acetylcholine opens the gates, the sodium quickly enters the muscle cell. This is known as the process of depolarization. Once you get enough sodium ions into the muscle cell, an action potential, which is an electrical current, occurs. Stimulus must stop, which means the action potential should stop.
This process is known as contraction.
When the action potential stops, the gates close. When the gates close, the sarcoplasmic reticulum releases the calcium it's been holding onto, causing the actin to change shape.
The length from left to right of the actin and the myosin is known as the sarcomere.
After calcium is released from the sarcoplasmic reticulum, calcium attaches to the actin, causing the actin to change shape for the myosin heads to attach. The myosin heads attach to the actin and carry the actin medially. This causes the H zone to get shorter, and this process is known as contraction. When ATP arrives, the myosin head is released from the actin. Now the myosin head combined with a different part of the actin molecule. The muscle cell shortens, which is known as the sliding filament theory. That means the bare zone is going away when the actin pulls together. When the actin is pulling together, the myosin also shortens. The A bands, which is the dark, is the part where it contains the myosin, and the I bands, the light, is the part where it does not contain the myosin. In order for the muscle cell to relax, the myosin heads need to detach. The stimulus must stop, and ACHE, an enzyme that breaks down, will occur. The gates will close on the muscle cell, and the calcium is reabsorbed by the sarcoplasmic reticulum. The actin changes back to its original shape, and now the muscle cell is relaxed.